here's someone who was not at Cambridge, Deb Kelly. She's in Seattle at University of Washington. And she discovered in the year 2000 this vent system called Lost City. These are both Lost City. They're called alkaline hydrothermal vents. And she was captain of the Alvin submersible, which has been discovered vents in the first place in the 1970s. This was a new type of vent that nobody had seen before. And this is the old style vent with called black smokers with a chimney that belches black smoke out. And this is a little bit of Lost City, and you can see that it's it's like a sponge. It's like a mineralized sponge that she's holding in her hand. So that's a sample from Lost City itself. So this is an environment that was proposed, in fact, 10 years before it was discovered by Deb Kelly. It was proposed to exist by Mike Russell. And after this discovery of Lost City, suddenly Mike Russell became quite famous. And this is taken from a feature article in Nature in 2009, where they photoshopped him up as Erasmus, the Renaissance man. And they called him Naissance Man, naissance as in the birth of life. He built a reactor at the Jet Propulsion Laboratory in Pasadena. And that's his reactor, and here's a little bit of Lost City behind him. Now, what Mike Russell was arguing 10 years before the discovery of these vents, and so it suddenly became very mainstream with their discovery, is that this is a kind of an electrochemical flow reactor, more or less. You've got reactive fluids percolating through there. We've got high concentrations of hydrogen in these vents. The early ocean's rich in CO2, and they would percolate in as well. And proton gradients, which is to say, the ocean's acidic, the fluids are alkaline, and they should mix in there over these catalytic walls that should contain iron, sulfur minerals, and things like that. So this is an electrochemical flow reactor that vaguely resembles cells. This is how it resembles cells. So we have here a bacterial cell. And it's got a pump in the membrane, and it pumps out protons. So this is the kind of thing I've been showing you. I've simplified it right down to a kind of ridiculous minimum. So there's protons outside, electrical charge on the membrane. Inside, it's relatively alkaline. And here's a pore in a hydrothermal vent, and it's alkaline fluids inside. It's acidic ocean waters outside. We've got a barrier. It's a lot thicker, but it's got some of the same catalysts in that barrier. How would you get from one to the other one? I'm not going to go through this. This is basically the work of my lab and other labs around the world over the last 10 years or more. But you'll recognize various bits of it. Here's the pore. We've got the protons outside. We've got hydrogen and CO2 and a reverse Krebs cycle making amino acids and lipids. Maybe those lipids can form a membrane. Then you can do a bit more chemistry. You can make sugars. So this is all the ungenetically, non-genetically coded metabolism that I'm talking about. I'm not going to go through the details of the experiments today. I want to get on to other more revolutionary things. And then I'm saying genes are arising in that context. So this is a hypothesis. There is evidence supporting some bits of it. There's lots and lots of work to do on this. But this will orientate you as to the kind of way you think about a question like the origin of life and the kind of experiments that you might want to do. I'd like to point this out, though, that the structure of a cell, which has basically got lots of hydrogen inside it, and it's got a negative charge inside and a positive charge outside, and it's pumping protons out, it's very similar to the structure not only of pores in these vents, but also of the Earth itself, which is full of iron inside, and you've got hydrogen bubbling out of these vent systems. It's relatively acidic on the outside. And so the structure of the Earth and the structure of a pore in a hydrothermal vent and the structure of a cell are kind of fractally similar to each other. So cells, I like to think of cells as little electromagnets that are resembling the Earth in their structure. And this kind of structure is probably very common. This is Enceladus. And there are plumes that were discovered by Cassini on the flyby of Cassini, these plumes jetting into space. So this is the icy surface. Enceladus is one of the moons of Saturn. It's got an icy surface. Underneath that icy surface, there's an ocean. And the reason we take it that there's an ocean is that these plumes are basically water coming from that ocean under some kind of pressure. And you can measure spectroscopically what's in those plumes. Well, it turns out they're alkaline, and they've got hydrogen gas, and they've got methane, and they've got various other various other organic molecules in there. There's no sign that there's necessarily life in there, but what this says is that the same kind of geology that gives rise to the hydrothermal systems on Earth are also happening on Enceladus, probably on Europa as well. And in fact, probably much more widely than that, because now we know, as you would have heard if you were here a couple of weeks ago, about exoplanets. There are something in the order of 40 billion, we would project 40 billion exoplanets, Earth-like exoplanets, wet, rocky planets and moons in the Milky Way alone. This is an extrapolation based on the number that have been detected over how many stars. So there could be a huge number, 40 billion wet, rocky planets in the Milky Way. They will tend to form these same kind of alkaline hydrothermal vent systems that we see on Earth and we see on Enceladus almost certainly. They will have natural proton gradients across cell-like pores, and those gradients will drive the reaction between hydrogen and CO2 to make the basic organic building blocks of life. And those should be the same everywhere. It's the same chemistry. And that also means that life elsewhere should face the same constraints that life did on Earth. We're back to this. The constraints are the constraints on bacteria, the constraints of just being a single cell. How do you get out of that? Well, I said it was an endosymbiosis. One cell gets inside another cell. What's the evidence for that? This has changed considerably over the last 10 years or so with the discovery. This is a hydrothermal system again. It's called Loki's Castle, and it's in the northern Atlantic between Norway and Greenland. And there was some archaea discovered in the slime underneath there, which became known as Loki Archaeota, after Loki's castle. And there have been quite a lot more have been discovered since then, and now finally cultured, and they're pretty hairy beasts. So now we have a whole Asgard superphylum. There's the Heimdall Archaeota and the Thor Archaeota. There's a whole Asgard nest of them. And these are the most closely related cells to eukaryotes as we know them. These are almost certainly something like this was the host cell that acquired a bacterial endosymbiont that went on to become the mitochondria. And the bacteria that were acquired, we don't know what they were exactly, but probably something like this. This is Rhodobacter. And this idea that life arose through a network of symbioses goes back to Lynn Margulis, also in the 1960s, another great pioneer of biology. And she had a vision of the planet as a symbiotic planet, that there were symbioses everywhere that you looked, and that the Earth was a system made up of a tapestry of symbioses. And you can see that this had some similarity with James Lovelock's Gaia hypothesis, the idea that the planet is in some way living. Now, Lovelock had originally put that idea forward almost literally as a living planet. He backed away from that idea, but his ideas live on in what we would now call Earth systems science. So this is a very dominant view in how we think about Earth history. And they published a number of papers together. This is a statue. This is actually in Lovelock's back garden in Devon, and this is a statue of Gaia that he had. His neighbor was William Golding, the novelist who had suggested the name Gaia to him in the first place. So it was really Lynn Margulis who had framed the idea that the eukaryotic cell was brought about by endosymbioses. She saw probably more of them than there actually have been in this history, but nonetheless it was a radical reconceptualization of how life evolved. And the bottom line on all of this is we've got cells within a cell, and here's the only example that we actually know about of a bacterial cell. This is in fact a cyanobacterial cell with bacteria living inside it. There's plenty of examples of complex eukaryotic large cells that have got bacterial endosymbionts or parasites or whatever, but this is the only known example of a free-living bacterial cell that's got other bacteria living inside it. So this, in my mind, is the starting point for where eukaryotes came from. And you can stare at it and think, well, why is that any good? Why is there somehow something better about that? And I think that it boils down to mitochondria have always retained their own genes. So these are mitochondria. This is in a single-celled protist called paramecium. Again, beautiful structures. These have tubular cristae, which are quite common in many protists. But all of these are individual mitochondria, and they all have their own copies of mitochondrial DNA. And it started out as a whole bacterial genome and got whittled away down to only the genes that are required effectively to control respiration locally. So each of these is a power unit with its own control unit inside it. If you want to scale up over orders of magnitude, and eukaryotes are on average about 100,000 times larger than bacteria, so if you want to scale up over that kind of scale, all you need to do is have 100,000 mitochondria, and you've done it. Really, that's it. You need to scale up your power packs. Now, that may sound crazy, but there are examples, a large amoeba have 300,000 mitochondria, so it's not actually madness. Is it real? Is it science, or is that simply an assertion? Well, it's a controversial area. I had a paper 15 years ago, which has been attacked by various people, for good reasons, no doubt, but I don't think they're right. I would genuinely like to stand in front of you and say, well, I got that wrong. And I don't think I did, though. Here's why. So this is a eukaryotic cell. This is the nucleus in blue here. This is where all our genes are. And the red dots and the green dots, so the green dots, let me get this the right way around, are the mitochondrial DNA, and the red is the mitochondria themselves. So all this is co-associated. The DNA goes inside the mitochondria. So you've got what I would call a genomic asymmetry. You've got all of these tiny power packs supporting energetically a massively swollen nuclear genome. And if you want to be, a complex human being, the genes that you have switched on in your brain and the genes that you have switched on in your kidneys or your liver or anywhere else, they're completely different genes. You need to have all of those genes in all of the cells in you, and that means you need a very large genome. You can't do that with a small bacterial genome. So what about a large bacteria? Here's one. This is called a Pulipiscium, and this is basically a giant vacuole in the middle. It's not a vacuole, but there's nothing going on inside that. These are DAPI staining of genomes, and there are 200,000 copies of the complete genome of that giant bacterium sitting inside there. If you add all up the cost of expressing all of those genomes, it's hugely costly. There's as much DNA in here as there is in there, and it will cost just as much to express all that DNA as this does. It's just that here we've redistributed it. We've reduced the overhead costs. So it's effectively we have multibacterial power without the overheads.